Hi, before we start, let me show you this picture. So this is just a gentle reminder that it's okay, it is alright to pause anytime or whenever you want. Hi, I'm back, and for this video we will be looking at how our immune system works. Basically, this is just an overview. Again, I've divided the topic into 5 subtopics. If you're familiar with this anime, Cells at Work, it's good. So, as we go along, I'll be showing some of the characters for those who are familiar with this anime, for them to be able to recall what these specific cells do. Okay, first part, let's look at immunity and its features and characteristics. Okay, so two objectives will describe the features of immunity, and we will look at the components of this immunity, which are your innate immunity and the adaptive immunity. Now, before we go on, what comes into your mind when we say immunity? Okay, now, according to Wikipedia, it's the capability of an organism to resist harmful microorganisms from entering. Huwag niya ko gayahin na, Wikipedia yung source. But, from our reference book, Phagocytic, so it states that immunity is the ability to resist almost all types of organisms or toxins that tend to damage the tissues and organs. Or, in other words, when we say immunity, it means defense or protection. So, it's a way of defending from harmful microorganisms or harmful things. So, for example, there's this boy. I'm sure you already know by now, since actually this topic is very timely since we are on a pandemic. Okay? So we should have an idea of what our immune system or how our immune system works. Just remember that our body, it is always exposed to a lot of harmful things. Take note, I'm saying harmful things, not necessarily microorganisms or live organisms. So this boy, he is always, let's say the body of this boy is always exposed to harmful things. And let's take, for example, this microbe with a crown. Let's not name it as a corona something. Okay? Let's just name it as a germ or a microbe. Okay? So in order for this, for the boy, in order for it not to be harmed by this microbe, okay, our body would employ a line of defense. So there are actually several lines of defense. So first one, there are barriers. And then we also have the foot soldiers, And we also have the tanks. And after some time, the special unit would come into play. Okay? We also have these special agents and a runner. And aside from that, there is a command center, which integrates all of the responses of these soldiers. So our immune system works like this, okay, in fighting off this crowned microbe or germ. Okay. Now, our immune system is like this. So we have the physical barriers, and then we have the innate immune system and the adaptive immune system. We will differentiate this as we go along. Now, some of the functions of our immune system would be the following. So it can be a defense against infection. So that's one of the primary functions of the immune system, infections, the defense against infections. It can also act as a defense against tumors. Okay. Aside from that, because of the process of inflammation, it can control tissue regeneration and scarring. Meaning to say, it can repair or initiate the repair of the damaged tissues. And then aside from that, it can also induce and injure normal cells. And it can also recognize and respond to tissue grafts and newly introduced proteins or anything that is not normally present in our body. Now, features of the immune system. So number one, there is what we call as your recognition. So of course, our immune system should be able to differentiate self from non-self, meaning to say our own cells should be differentiated from the non-self cells. Okay. So any foreign substances and organisms that have entered the body should be recognized by our immune system. We will discuss the major histocompatibility complex as we go along this lecture. So this is an example of how our immune system recognizes these foreign substances and organisms. So usually these foreign substances and organisms, these have what we call as your PAMP, okay, or your pathogen-associated membrane proteins. So when these PAMPs are then recognized by your PRRs, or the receptors that would recognize the PAMPs, okay, it would cause a cascade of events, okay, leading to either the death of the cell that would contain the PAMP, or this PAMP would be ingested by another immune cell. So this is the process of elimination, wherein a diverse repertoire of cells and molecules act in concert to neutralize the threat. So it would control the spread of this pathogen and these PAMPs. Another feature of our immune response is what we call as your immunological memory. So meaning to say, our immune system can learn from previous encounters with the microbe. So in other words, it would maintain a reserve of cells. So these cells would be called as the memory cells. So these are a reserve of cells that are able to respond swiftly whenever a new infection is brought about by that same microbe that was previously encountered. And we have what we call your proportionality, wherein regulatory mechanisms would exist to ensure that the immune response are proportional to the level of threat. So example, so for mild injury, okay. So this is, for example, this is a cell, and when that cell had a mild injury, it will initiate a cascade of events which is brought about by these DAMPs. DAMPs, on the other hand, these are endogenous proteins of the cell. These are called as danger-associated membrane proteins. So whenever the cell is injured, these DAMPs would be expressed, which results to cell death. or apoptosis. Now, this apoptotic cell will then be ingested by another immune cell, which is your macrophage. So, at this point, during mild injury, the immune system remains quiet. As compared to when the injury is severe, wherein there is an uncontrolled cell death resulting to the release of damps into the extracellular environment. So when the cell bursts or dies, the damps would be spreading into the extracellular environment. And then these damps, same way as with your pumps, this would be recognized by your PRRs, resulting to an activation of the immune response. Okay, so as we go along the video or lecture, please fill this up. So these are the different components of our immune system. We have the innate immunity, the adaptive, and the adaptive can be further subdivided into cell-mediated and humoral immunity. And kindly fill this out. So as I mentioned earlier, our immunity can be divided into your innate immunity and adaptive immunity. But take note, they are not independent with one another. In other words, one depends on the other, or they are interdependent with each other. So these are the components of our innate immunity rather. And for the adaptive immunity, for the cell-mediated, this is a function of your T lymphocytes, and the humoral immunity is a function of our B lymphocytes. In terms of the action time, this is how the innate immunity is different from your adaptive immunity. Since the innate immunity is non-specific, it is always there, it does not need time to be activated, unlike your adaptive immunity. Its action time is very fast. So usually at the onset or at the exposure to the injury, or having the injury or exposure to the microorganism, the very first responders would be the innate or would be the components of the innate immunity. Now after some time, that's when the adaptive immunity kicks in. So usually this would take a few days before your adaptive immunity becomes active. Okay, so we're done with part 1. This is a reminder to stretch. Okay, so you can pause and stretch. Okay, after stretching, let's now proceed. Okay, second part would be the innate immunity and its components. Okay, so this would be the objectives for this part. So we will look at the different components of the innate immunity. And then last part of this video is the regulation of the innate immunity. For the innate immunity, so this, as I've mentioned earlier, this is an early defense against infections or injury. Please remember that our immune system is not exclusive to infections, or rather, our immune system would be activated or can be activated if there is an injury. Now, the injury can be either a physical injury, chemical injury, what else, or an infection. if it is caused by a microorganism or toxin. Now, the defense is brought about or because of several small set of reactions. However, the problem with innate immunity is that it is more limited compared to that of the adaptive immunity. Okay, so these are the features of innate immunity. So number one, it is still specific. So even though it is termed as an innate immunity, it is always there, it is still specific, meaning to say it can differentiate self from non-self through these receptors, your PRRs, or your pattern recognition receptors. One classic example of these pattern recognition receptors would be the toll-like receptor, which is the one found on the surface of macrophages. The distribution of receptors is non-clonal, meaning to say it is identical for all cells of the same type. And as I've mentioned earlier, it can discriminate self from non-self. Now for the components, so you have the barriers. These are your skin and the mucosal lining, or the one that lines the mucosa. You also have the phagocytic cells, your neutrophils, macrophages, and eosinophils. We also have your natural killer cells, inflammation, or the process of inflammation, complement activation, and cytokines like your interferons. For the barriers, so let's look at the skin. So this is your skin, for example. So present on your skin would be your keratinocytes. So take note, your keratinocytes, aside from producing keratin and aside from lining our skin, forming a strong physical barrier, it would also express PRR. So meaning to say, it can distinguish foreign from the self cells or non-self cells from the self cells. Aside from that, you also have microbicidal agents produced by your sweat glands. Okay. So example of this would be a very low pH, acidic pH, and factors such as your defensins, which would prevent the growth of microorganisms. You also have your commensal microorganisms. So these commensal microorganisms are present in our skin without causing any harm. And these commensal microorganisms would inhibit the growth of pathogenic or harmful microorganisms. And there are other immune cells present in our skin. Of this, please take note of the Langerhans cell would phagocytose microorganisms, and it can process the non-self proteins. Okay, for the mucosa, this would line the surface of the gastrointestinal tract, genitourinary tract, and the respiratory tract. So mainly this. So what is special about this mucosa is it has a lot of secretions. And what can be found in these secretions would be several factors. So number one, you have a very low pH. Example would be the gastric mucosa, wherein there is hydrochloric acid. So the pH would be very low, which prevents the growth of majority of microorganisms. Aside from that, in other areas, there are proteolytic and microbicidal factors. And for the respiratory lining, we have mucus, which more or less acts as a trap for the microorganisms. here. So these are the mucus. For the gastrointestinal tract, in the esophagus, we also have mucus. So one example of how these factors work as a protection would be the mucociliary clearance. So I will give you the link for this video. Okay, now let's look at the components or other components of the innate immunity. We have the phagocytic cells, and I think this was already discussed in your WBC lecture. Okay, so just a review. So some of the phagocytic cells would be the macrophages, so either the free or the one roaming around your blood, the one found in your blood. And you also have the fixed macrophages, which are specific on each organs. Okay. You also have your neutrophils. We also have your neutrophils and your eosinophils. Okay. Just take note that, as I've mentioned earlier, one important PRR or pattern recognition receptor for this one would be what we call as your TLR, or the toll-like receptor. So this toll-like receptor is a receptor found on the surface of phagocytic cells that, when it comes in contact with a foreign substance or a microbe, for example, it will start phagocytosing it. We also have the process of opsonization, wherein certain factors would coat the microbial cell, and once coated, there would be an enhanced phagocytosis. So it more of acts like it increases the likelihood of phagocytosis of this microbial cell. It makes the microbial cell more palatable to the phagocytic cell. We also have your nets, your neutrophil extracellular traps. So this is primarily used by neutrophils and eosinophils in trapping larger microorganisms like helminths or worms. And we also have the process of inflammation. So again, I'm sure this was already discussed in your WBC lecture. So just remember the chemical mediators for inflammation. So this is again an overview of the inflammatory response. Okay. Now let's look at another cell, which is your natural killer cells. So this is an example of a natural killer cell. So it's a little bit different from your neutrophils. So it is not multinucleated. It is not multinucleated. It has a single nucleus only. So this is a small population of lymphocytes. So take note, your natural killer cells are a small population of your lymphocytes, specifically the T cells. So this would express your CD8 proteins. We'll discuss these CD proteins as we go along in your cell-mediated immunity. Now this would use your natural killer receptors or NK receptors, okay, which is somehow different from other lymphocytic receptors. Now, the main purpose of your natural killer cells is it would kill cells, from the name, natural killer. It would kill cells that would abnormally express the MHC. Once the NK receptor is activated, okay, it would secrete your interferon, one of the cytokines. Okay. Now, this interferon would activate other immune cells, specifically the macrophage. There are several mechanisms by which the natural killer cells act. So before we proceed, this is natural killer cells from Cells at Work. So she's deadly. And take note, in the anime, she uses a sword. So you'll know later on why there's a sword. So the three mechanisms would be the death receptor pathway, the granule-dependent pathway, and the antibody-dependent cellular cytotoxicity. So this is how NK cells act. So it can either directly kill an infected cell. So the NK receptor recognizes this abnormally presented MHC. Okay, so it would kill the cell, or the NK cell can secrete interferons, which would activate other immune cells, and then these immune cells would be the one killing the cell. This picture shows us the different mechanisms by which the natural killer cells act. So number one, there is the death receptor pathway. So in your death receptor pathway, take a look at this one. So on our natural killer cells, there is what we call as your fast ligand. Here, fast ligand. Okay. So this fast ligand is recognized by the fast receptor. Now the fast receptor is located on the target cell or on infected cells. So this would be expressed if a cell is injured or infected. Now once this is activated, it will result to a cascade of events resulting to caspase activation. Now remember, when we say, anyway, you will learn more of this in pathology. Now just remember that when we activate our caspases, it will ultimately result to apoptosis or cell death, programmed cell death. The other type of mechanism by which your natural killer cells work is through the granule-dependent pathway. So in your granule-dependent pathway, the natural killer cells would secrete its granules. Now located on these granules would be two factors, what we call as your granzymes and perforins. Now the perforins are like proteins that would insert itself, or that would be inserted and open up the membrane of the target cell. So it's like it will open up a tunnel into the cell. Now once the tunnel is opened up, the other factor, which is the granzymes, would enter the cell. And then these granzymes, these are hydrolytic enzymes, hydrolytic or proteolytic enzymes. So this will digest the organelles of the cell. And same way, it will also induce caspase activation and eventually leading to apoptosis. So this is the reason why the natural killer cell in Cells at Work has a sword, because it pierces through the cell membrane of the target cell. Now the other mechanism of your natural killer cells is through this one, which is your ADCC, which is the antibody-dependent cytotoxicity, wherein the natural killer cell would recognize an antibody. This is the antibody that is bound to a cell or to a microbe. Now, once it recognizes this antibody, the natural killer cell will attack the infected cell. Another type of cell would be the dendritic cells. Now, what is special about these dendritic cells is that they are also phagocytic cells. Okay, But instead of breaking down the ingested protein or microbe, what this dendritic cell will do is it will migrate to the nearest lymph node as an antigen presenting cell. Sorry, there is a typo error. This is antigen presenting cell, which will be discussed in adaptive immunity. So here, so this is an example of an immature dendritic cell. So it's non-motile, it does not move, but it is highly phagocytic. It will continuously phagocytose anything in the environment. And then if it encounters certain factors, for example, this cytokines or a PAMP, pathogen-associated membrane protein, it will be activated. It will now become like this, a mature dendritic cell. Now, your mature dendritic cell, it is highly motile. It needs to move. Or in other words, it needs to migrate to the nearest lymph node in order to present what it has ingested to the lymphocytes. So this is dendritic cell from Cells at Work. Okay, so this is a meme of dendritic cell. Okay, another reminder for stretching. Okay, so you can pause, stretch, and be back after a while. Moving on, next component would be the complement, or the complement system. Now, these are a set of plasma proteins that are activated in a cascade-like manner upon binding to certain factors that are not normally present. So they are initially thought as a complementary to the antibodies in fighting off an infection. That's why it's called the complement system. However, please take note that the complement system is actually a part of the innate immunity. And please answer this, why? Why is it a part of the innate immunity? Now, there are several mechanisms how the complement system helps in fighting off an infection. So you have your opsonization, inflammation, and cytolysis. I think you already know number 1 and number 2. Again, opsonization, this one makes these are the factors, or opsonins are the factors that would coat the microbial cell, making it more likely to be phagocytosed by the phagocytic cells. So before we proceed to the different pathways on your complement system, please take note of this, your cascade activation. So usually the complement proteins are designated by letter C, uppercase letter C, and a number. For example, in this case, C1. And they are inactive, this one. Now, whenever it is activated, or whenever it is cleaved off to become an active factor, the active products are usually designated as an A or a B. So, for example, C1A and C1B. So, this will be the factors produced if we split or if we activate C1. Okay, so are you guys ready to look at the complement system? Now, there are several pathways that used to activate the complement system. Okay, we have the classical pathway. So take note, this is dependent on the specific immune system, or in other words, the adaptive immune system. And then we also have the alternative pathway, which is a non-specific, it depends on the innate immunity. And a third pathway would be the lectin pathway. Now, in the classical pathway, so take note of the trigger, or the one that starts the cascade. It is triggered by the binding of the antibodies to the microbial cell. And then the endpoint of this is the cell will die, or the microbial cell will die. Now take note that the antibodies here are a component of the humoral adaptive immunity. Now remember the 3 A's: the activation, amplification, and attack. So this is an illustration of the classical pathway. So we have an antibody that binds to the bacterial cell wall or bacterial membrane. What happens next is the C1 protein will bind to it. The C1 then would cleave off C4 and C2. Okay. Now take note of this. The C2A will form a complex with C4B, forming now what we call as your C3 convertase. So the C3 convertase, this would now cleave C3 into C3A and C3B. Now take note of this product here, C3A. So C3A, this is considered an anaphylatoxin. When we say anaphylatoxin, it increases the process of inflammation. How? Okay. So your C3A would bind to receptors located on the basophils and mast cells, which would result to release of its granules or vesicles containing histamine. So this histamine, these are vasoactive compounds, so this would enhance vasodilation and permeability. So more inflammation. Now, if you look at C3b here, it will again form a complex with. Now, so if you look at C3b here, the other product, when you hydrolyze C3, it will now form a complex with the C3 convertase. Okay. Forming now C5 convertase, which cleaves C5 into C5a and C5b. C5a, again, this is another anaphylatoxin, so it enhances vasodilation and vascular permeability. And aside from that, this is a very strong chemotactic factor for neutrophils. So if you can remember chemotaxis, the process of chemotaxis, one of the factors that initiate the neutrophilic migration to the site of injury would be C5a. So it sort of calls more of the neutrophils into the site of injury or infection. Now C5b, on the other hand here, will bind to the membrane. And together with other complement proteins C6, C7, C8 will form your C9, which is what we call as your MAC, or the membrane attack complex. So this is a protein complex that inserts into the microbial cell membrane, resulting to lysis. So this is somewhat similar to the perforins of the one used by your natural killer cells. So this would open up holes into the membrane of the microbe. So like that. This is again a meme showing us the membrane attack complex. So this is a picture of a bacterial cell that has lots of MAC attached on its membrane. So it's like it has a lot of craters in it. Now, when you have these holes on the microbial membrane, okay, of course, water and other ions can enter into it, resulting to bursting of the cell. So that would be for the classical pathway. For the alternative pathway, okay, this is a non-specific defense because it does not rely on the antibody binding to the microbial cell wall or cell membrane to initiate the pathway. And this is slower, a little bit slower than the classical pathway. And as I mentioned earlier, it does not require recognition of the antigen. But what happens here is that there is spontaneous conversion of C3 into C3A and C3B. Okay. Now take note that normally, or in the absence of a microbial cell, okay, C3b is very short-lived, and this is quickly inactivated by other proteins on our own cell membrane. Take note, this is cell membrane, not cell wall. However, in the presence of a microbial cell, so when this C3b binds to the membrane of the microbial, of a bacterial cell, for example, it does not have these surface proteins that would hydrolyze or inactivate this C3b. So in other words, C3b stays attached to the membrane of this bacterial cell. So this is an illustration of the alternative pathway. So there's a spontaneous hydrolysis of C3, and in the presence of a bacterial cell, C3b tightly binds into the membrane. We also have a C3a formed here. Now C3b will now cleave factor B, or protein B, okay, together with protein D, forming BB and BA. Now take note that this BB will now form a complex with the C3b, forming now your C3 convertase, which would now cleave C3a into C3a and C3b. So you have more of the C3a being formed, and you also have another molecule of C3b. C3b again would form a complex with the C3 convertase, forming your C5 convertase, which would then cleave C5 into C5a and C5b. Again, you also have another anaphylatoxin and chemotactic factor formed here in the form of C5a. C5b, again, same as with the classical pathway, it would form a complex with C6, C7, C8 to form your membrane attack complex, which is your C9. So both pathways, the end result is you produce a membrane attack complex. Now, the lectin pathway, on the other hand, this is activated in the presence of your mannose binding lectin, which are usually present in the cell wall of bacterial cells. So in the presence of bacterial cells, this lectin pathway can be activated as well. So it would this lectin would activate the MASP, your mannose activated serine proteases, which would cleave or hydrolyze C2 and C4 into C2A and C4B, forming a C3 convertase. And at this point, this is where it will merge with the classical pathway. Okay, so in summary, this is the different pathways for complement activation. So classical pathway, antibody binding to the bacterial cell, which initiates the formation of C3 convertase. Same way as with your lectin pathway. So you have your mannose binding lectin, which activates the serine proteases, which also activates your C3 convertase. The alternative pathway, you have spontaneous cleavage of C3, and together with factors D and factor B would produce your C3 convertase. C3 convertase would then cleave C3, and together with C3b would form your C5 convertase, and so on and so forth until you form the membrane attack complex. Again, regardless of which pathway, you produce several things or several factors. Number one, you produce the chemotactic factor, which is your C5a. You also produce your anaphylatoxins, which are your C3a and C5a, which would enhance inflammation. And lastly, you would also produce the membrane attack complex, which is this one. Now, in terms of regulating the complement system, so there are several factors that would inhibit the spontaneous activation of the complement proteins. Number one, you have your factor 1, which would inactivate the C3b. Take note, this should be small b. You also have factor H, which works in the alternative pathway by removing the BP protein, which would prevent the formation of your C3 convertase. And you also have the C1 inhibitor for the classical pathway, which would inhibit the binding, or rather the activation of C2 and C4 from your classical pathway. Another component of the innate immunity would be the secretion of cytokines. Okay. So these are soluble proteins that would act as immune mediators and inflammatory mediators as well. So in other words, these are chemical messengers which acts as a communication between leukocytes and other immune cells. And take note, this would play a role in both the innate and adaptive immunity. So here, these are the examples of your cytokines. So you have your interleukin 12, interferons, TNF, and your interleukin 1. So there are actually a lot of cytokines. Okay. So we can classify your cytokines as inflammatory or pro-inflammatory cytokines. So this would promote inflammation. So go inflammation. Examples would be your TNF alpha, interleukin TNF alpha, okay, secreted by the macrophages. Interleukin 1 also secreted by macrophages and other phagocytic cells. and your interleukin 6 here. So each of these primary cytokines, pro-inflammatory cytokines, would have different effects. And we also have your anti-inflammatory cytokines, which would stop or reduce inflammation. Example of this would be TGF, this one. So this is primarily an inhibitor of inflammation. Okay. What else? Interleukin 10. So in reality, our immune response is actually a balance between the pro-inflammatory cytokines and the anti-inflammatory cytokines. Now, one of these cytokines would be the interferons. So these are produced by infected cells, either viral or bacterial. Okay. Now, take note that the type 1 interferon primarily acts as an antiviral agent. Okay. So this is secreted by infected cells or immune and other immune cells, but primarily this is secreted by your dendritic cells. Okay. Now, the main function of your interferons is it activates other immune cells. Example of these immune cells would be the natural killer cells. So this one. So this is an example of an infected cell, virally infected cell. So this virally infected cell would produce your interferons. Now this is together with your dendritic cells, would also secrete your interferons. Now these interferons would be recognized by other adjacent or neighboring cells, and then once it is recognized, it will result to a cascade of events resulting to production of proteins that would block the entry of the virus into that cell. So it's sort of like it prevents, or the virally infected cell sort of tells the other cell that, hey, there's a virus, you need to produce these proteins so that the virus cannot enter. We also have the acute phase reactants. So these are plasma proteins that act as early alarm mediators. So usually this would increase in concentration in response to an injury. So these are examples of the acute phase reactants. Now, in the regulation of our innate immunity, this is to prevent excessive tissue damage because it's non-specific, okay, unlike the adaptive immunity. And this is through the secretion of anti-inflammatory cytokines. Example, as I've mentioned earlier, you have your interleukin 10, which inhibits the macrophages. You also have the secretion of interleukin 1 receptor antagonist, which prevents the function of the interleukin 1. And you also have feedback mechanisms. So this is an example of a feedback mechanism wherein the inflammatory state would signal the immune cell to somewhat stop or slow down in its function. So in summary, what we have discussed so far would be, number one would be the features of immunity. These are the recognition through your receptors, pattern recognition receptors. You also have memory. You also have elimination and proportionality. We have also looked at the differences of the innate and adaptive immunity. However, we will look more on the subsequent videos. And we have looked at the components and function of the innate immunity.